This is Cognitive Neuroscience. Hope you guys are all in the right spot. It's like 4:49 and Taylor got 3. So consider for a moment the fact that each and every one of you is composed of billions of cells, right? And this isn't even taking into account the billions of cells that aren't you, right? Now, each and every single one of these cells contains the exact same genetic material, the same blueprint for developing and manufacturing proteins. Yet, despite the fact that every single one of these cells has the exact same blueprint, we have hundreds of different kinds of cells, different shapes, different sizes. Each of these cells has a specific and a unique function that kind of benefits the organism as a whole. And these cells, they're living entities. They maintain their own homeostasis. They create their own energy. They have metabolic needs that need to be met or else they'll die. And these living things, these cells, they organize themselves into communities that we call tissues. Right? And these tissues kind of mingle and merge together, and they form these like heterogeneous communities of living things that are able to achieve these incredible cooperative functions like pumping a heart or drawing in air to the chest and absorbing oxygen, feeding the rest of the body. And to add to this layer of complexity, these organs are further organized into these incredibly complex systems, right? Where you have communities of cells coordinating with each other and communicating with each other over vast biological distances, right? And at the center of it all is the nervous system, collecting information, checking in and making sure that everything is running smoothly and everything is running efficiently. The nervous system knows what's going on in your body and its community of cells at every given moment. And it also knows what's going on outside of its own community of cells. It can move its cells through the world to collect more resources. Right? It can move its cells to protect its community of cells. And at the very tip top of all of this complexity is what we call consciousness, right? Unaware of billions of interactions that are happening every single second, right? All consciousness has is like the headlines, right? It's like a CEO of a company or like a ruler of a country that's just kind of talking to its advisors, right? Like, how's the foot doing? The foot's doing good. We're good. Are we hungry? Yeah, we could eat something. All right, send some commands to go find food, right? We feel like we're so aware of everything that's going on at any given moment. It's intuitive, right? We feel like we have so much control over our actions and our thoughts and our desires. But really, just like when Galileo was arrested for trying to claim that the Earth wasn't the center of the universe, science today, brain science, cognitive neuroscience, is really causing us to question our place in our own reality. This like immense self, this sense of self that we have is really at the fringes of everything that's going on. I mean, there's billions of things that are happening every moment that it doesn't know about. And the brain, its ability to do this is incredible because the brain doesn't see light. It doesn't hear sound. It doesn't touch things. It doesn't smell or taste things. It's locked away in this protective dark skull, and all it gets are electrical and chemical signals. And somehow it's able to turn all of those signals into what we experience every day. So throughout this class, we're going to talk about how the nervous system collects that information, how that information flows through the system, and how the brain ultimately uses that information to create these amazing perceptual experiences that we have every single moment of every day. Right? So we'll start with kind of talking about what cognitive neuroscience is. It's kind of a loaded term. There's two halves to it. So the cognitive side of it comes from philosophy and from the cognitive revolution in psychology. It's this attempt to try to understand the brain as an information processing system, right? And cognitive psychology, when it got its start, didn't have access to the technology that we do now. And something I really want you guys to kind of put into perspective and not take for granted is that cognitive neuroscience is an extremely new discipline. I mean, we're talking the last 20 years that most of this stuff has happened. The beginning of cognitive psychology actually used things like reaction time to try to figure out how the brain was doing things, right? It was looking at how long it was taking to process things based on how long it was taking people to respond. Now, neuroscience, on the other hand, is a biological discipline. It's a discipline that tries to understand the biological underpinnings of how the nervous system works, right? The neurons themselves, the cells, the molecular interactions that are going on between these things. And cognitive neuroscience is more macroscopic, right? If I just say neuroscience, if I'm talking about a neuroscientist, usually I'm referring to someone that does molecular work, someone that does studies with animal models like rats and mice and monkeys, and athlesia and zebrafish, things like that. The cognitive side of things was saying, okay, there's a lot in animal systems that we can't figure out, right? Animals can't tell us about their perceptual experience. But now we have these amazing technologies like MRI and EEG and all of these new ones that are coming out that allow us to actually see what's going on in the brain while we're thinking about all this stuff. So just kind of an overview of what we're going to be doing in the class. We're going to start today with sort of a history of the discipline, kind of put into perspective how new this is and how we really didn't know much about the brain until like the last 100 years. Like it's really cool that a lot of people kind of take for granted. We didn't even know that like neurons were a thing until the 30s, right? And then in the next lecture, we'll go on kind of a brief tour of the nervous system, of the brain. That one's going to be kind of a quick lecture. There's a lot to get through, but it's something that's going to just be kind of the seed. I just want you guys to know about all the pieces of the puzzle, and then we'll kind of work backwards. And we're going to start with early sensory processing, right? Try to figure out how the body itself, how the nervous system collects all of this information about the world, and then how that information is processed in these very specific regions. These primary regions are just processing one sense, right? And then the information from these primary regions is then distributed and shared throughout the brain. And those are the areas that we'll talk about later in the term when we get into higher cognitive processes like attention, learning and memory, language, things like that. So just give you guys a heads up, this class is very information heavy. I was kind of hinting with people before class started, 10 weeks is not a lot of time. Cognitive neuroscience is a gigantic field. There's 20,000 MRI publications every single year. That's like way too much to get through. But we're going to do as best we can. If I am going too fast, if there's a lot of information to get through, I am actually recording all of my lectures, and I think I'll get into that in a minute. And something that I'm doing, I don't take attendance. I don't believe in requiring you guys to be here. You're all paying to be here. And honestly, I'd rather have people here that want to be here. And so I've designed my slides as study guides. So as we're going through class, the slides that I post on Canvas are incomplete. There's blanks in them. And those blanks are very specifically placed to try to guide you guys towards the information that I think is important. There's a lot of stuff that we'll cover that is contextual that I don't want you guys to memorize. I hate this like form of academia where you just like memorize and regurgitate a bunch of information and then it's gone. So this will help you kind of like focus where you're studying. And I also am going to be recording screen capture and audio of all the lectures that I'll try to share with you guys. So if I am going too fast, you guys will be able to re-access the lectures, kind of take notes from there. So we're going to go into a kind of perspective of the history of what led to kind of where we're at right now. Like I said, this is a very new discipline. It's like 20 years old. So we're going to look at some of the precursors that led to what we're able to do now. And so we're going to start way back with how the brain evolved, right? And then we're going to see how humans started to think about what the mind was and what the brain was doing. And then kind of turn of the 20th century, we'll start looking at these pioneer neuroscientists that started the molecular revolution in neuroscience, which is huge. Like the neuroscience field in general, cognitive neuroscience aside, is a gigantic field. Society for Neuroscience conferences are like thousands of people big. There's a lot of work being done on the molecular side. So we'll end today's lecture with kind of the early steps in cognitive psychology, and we'll end with kind of where we're at now and what tools we're using to kind of figure out where they were missing things in cognitive psych. Cool. So we're going to start with the evolving brain. And so you guys have probably heard the term lizard brain before. And what people are referring to when they say things like that is the phylogeny, the phylogenetics of brain development and evolution. And phylogenetics is actually the study of how structures are conserved through evolving organisms, right? And so what you can see here in this picture is that as the organism got more complex, the same structures were kind of maintained and then built upon, right? And so when someone says lizard brain, they're actually talking about the part of your brain that's very evolutionarily old. And these older evolutionary structures, they cover a lot of the really basic functions of life, right? Pumping the heart, making sure the lungs are working. You think about really simple, complex organisms. That was kind of an oxymoron, but they're not social creatures most of the time. Like alligators don't have friends, kind of things. Those are things that higher brain areas were able to accomplish. And so these older brain structures, when you're looking at like MRI images and stuff, this is a mid-sagittal slice, so this is right through the middle of the face here. And it's showing that a lot of these older structures are buried really deep in the brain. So you have like medulla and the brainstem here and the pons, thalamus, and then it built on top of that. And so you have the next layer of complexity is emotional processing, the limbic system that we'll get into. And then the highest form of cognition comes from the cerebral cortex, which allows for a lot of flexibility with the information that's being processed. So if we really try to put the evolution of the brain into perspective, we've got to think about these like incredibly big numbers for a second, right? So the primate brain was 34 to 23 million years ago. And then the great ape, which starts to get into chimpanzees and gorillas and things like that, that evolution happened 23 to 7 million years ago. And it wasn't until about 7 to 5 million years ago that we started to see the first Homo sapiens, right? Now, put these numbers into perspective, right? Cell phones have only been around for 30 years, right? The way that our society is organized, the country we live in, is not very old. I mean, 5 to 7 million years is a lot bigger than 150 years, right? And so you really got to keep all of that in perspective when you're thinking about why the brain does what it does, the type of information that it's using, right? Because the brain evolved in a hunter-gatherer type setting, right? So a lot of these functions developed to fulfill those roles. So as time went on and the human brain got more complex, people started to think about where their experience was coming from, where the mind was located, right? And the earliest recordings that we have of this were about 800 years BCE. And this was like the early Homeric Greeks. They knew that we had this ability to have perceptions and different types of mental experiences. We're mad sometimes, we're sad sometimes. They thought that this was because of multiple souls that were associated with different parts of our body and different aspects of mentality. And as time went on, about 500 years before our 0 point, we started to see these two camps emerge of saying, you know what, I think it's a lot simpler than that. I think we can kind of locate where the mind is at. The first group was cardiocentrists. And these guys believed that the mind was in the heart. And this is a really intuitive kind of idea, right? I mean, if you don't know anything about anatomy, if you've never cut anybody open and looked at a brain or anything, the only thing that you can really feel moving is your heart, right? It's kind of this intuitive sense that that's what's creating life. And when the heartbeat stops, everything else stops. But then this other group of Greeks started to say, you know what, I think that a lot of these mental faculties are actually localizable to the head. You had this guy, Alcmaeon of Croton, who discovered nerves when he was like dissecting these animals and noticed that the nerves were sending information to the brain. And you had Hippocrates, who was way ahead of his time, that actually noticed that brain injuries were causing differences in mental abilities, right? He said, you know what, I think that the mind is up in the head where all of this stuff changes when we hit it. And the next slide, you'll notice, skips a lot of time. A lot of this was due to the Dark Ages, of the burning of the library in Alexandria. Like, I wonder what would have happened if the Greeks had continued their scientific pursuit like 2,000 years ago. But the game really starts changing. And there was a lot of people involved before Descartes, but Descartes gets brought up a lot in terms of brain science because he was a big pioneer in changing the way that we were thinking about things, because we're looking at the 1500s now, right? So we have this whole chunk of period where Christianity started to dominate Europe. And there was this belief that the body itself was divine, that all of our experiences were divine. And Descartes started to notice that he could control the body without he could stimulate nerves and it would move things, right? So he started to develop this idea that the body is really just a machine that's being controlled. And he had to grapple that idea with his religion, right? And trying to understand how to put these two things together. And so his idea was that the body itself is a machine that's being controlled, but it's being controlled by something exterior. And this is this idea of dualism, that there's these two separate interacting things. And this idea that he was searching in the brain for where this like location was that the soul was like interacting with the body and exerting its control. And he came to the conclusion that it was the pineal gland, because the pineal gland is the only non-bilateral structure in the brain. And this was a really, really big turning point in history. I mean, this idea really swept and took over and was extremely dominant for 3 centuries. But we started to kind of work out this idea through what Hippocrates was kind of getting at in like the early Greek stuff, that if we damage the brain, it's also damaging function. And it started to look like maybe it's the flesh and blood, maybe it's the system itself that's producing this phenomenological experience, right? And this is this idea of monism, that there's not these two separate interacting things, but the body itself is creating this perceptual experience that we have. And Thomas Willis was about 100 years later, and this was around the time where a lot of dissections were being sanctioned by the government. They wanted to know more about anatomy. And so they were actually having criminals dissected by these local doctors to figure out more about anatomy. So the king of the area would like give these scientists right to all of the people that were hung to dissect them and figure things out. Well, there was this lady, Anne Green. This is a really kind of interesting story. She was charged with infanticide. She had a stillbirth baby. And back then it was just this idea that it was her fault. And she was condemned. She was brought to the gallows. She was hung. And after she was hung, her body was sent to Thomas Willis to do a dissection and to figure things out. And while she was laying there on the examination table, Willis actually noticed that her eyelids were flickering and that she was still alive. And he nursed her back to health. And there was this big controversy at the time because people wanted to hang her again. They wanted to try her again. And Willis actually came to her defense and argued that it was God's will, that she was like saved, and they put on this huge trial. It was this big event, and ended up that it saved her life, and it also propelled Willis into this like really popular position that gave him a ton of funding. And that funding was really important because Willis was one of the first people to start dissecting the brain and start identifying structures. And you got to think about this, right? A lot of these structures, if you were to take an anatomy class and they were to have you like memorize all of these brain structures, a lot of those brain structures were named really, really early on from these dissections, where they were just seeing like differences in shapes on the brain. They weren't locating things based on function or anything like that. It was all about the shape of the brain. But this is really cool. There's a lot of stuff that he named way back in the 1600s. If you've ever heard of the Circle of Willis, the blood circulation in the brain, that was actually named after him because he's one of these like first pioneers in brain science. And then along came Franz Joseph Gall, which I'm sure you guys have all heard about in intro psych and things like that. And around this time when Willis started to identify that there were these unique structures in the brain, it kind of propelled this idea that if there's different structures, then maybe different types of faculties are localized to those different structures. And so Franz Joseph Gall started the practice of phrenology, which was this idea that we could localize very specific cognitive functions to these different areas. Now, this idea of localization is something that's going to come up a lot throughout this course. But I want you guys to be really, really clear about the fact that he got it really, really wrong, right? Look at these 35 functions that he defined, right? These are very complex functions. We have noticed, and we'll talk about how there are very specific brain structures that process very specific types of information, but it's not complex information like this. Complex cognitive abilities require diffuse activation. They require lots of cooperating regions and things like that. And he argued at the time that if we used a part of the brain more, that part of the brain would get bigger. Just like if we were flexing our muscle, if we were working out. He considered the fact that the brain was a muscle. And this is somewhat true. There is a hint of truth to that. There are some regions like the hippocampus that will actually get bigger for people that are memorizing lots of things. But there are also parts of the brain that bigger is less efficient, right? So there's definitely trade-offs. This idea that he believed he could measure all of this on the skull is complete nonsense. And this actually started a lot of really bad practices. A lot of people were put down. This was a big thing in slavery, of trying to claim that they were where they needed to be because of the shape of their skull. What you need to know about him is that he was not a true scientist in the way that we think about it. He used correlational research, which is not a bad thing. fMRI, as you'll learn, is correlational research. But what he was doing wrong was he was trying to use those correlations to confirm hypotheses that he already had. He was saying, I believe that combativeness is behind the ear, and I found someone with a bigger part there that's combative, and so that means that's where it should be. And that is not the way that you should be doing science. But this idea really took off. It was very intuitive. And it was like all over the place. People going in to have their heads measured to see if they were like getting married to the right people. They were trying to justify why certain people were leaders and certain ones weren't. I mean, this is ridiculous. Like, chins show how firm you are. Like, I mean, it's even going on still today, right? And these two phrenologists came to very different conclusions. Like, but what kind of came out of the stuff around phrenology, and this is kind of an interesting story. So Franz Joseph Gall was brought to France to measure Napoleon Bonaparte's skull. And Napoleon Bonaparte kind of considered himself to be this like amazing and like supernatural being, right? And Franz Joseph Gall measured his skull, was like, this is where you're at on all these things. And he wasn't a good politician because he didn't tell Napoleon that he was special. And Napoleon was like, you're full of shit. And I'm going to prove you're full of shit by hiring this guy, Pierre Florenz. And Napoleon sent this guy to try to disprove Franz Joseph Gall. He said, I want you to show that what he's doing is crap. And so what this guy Florenz did was he started to work with pigeons and rabbits, and he started to destroy certain parts of their brain to see how it affected their overall behavior, right? And what he noticed was that there were specific localized functions, like I just mentioned before. But it was very basic type stuff, coordination, things like that. He couldn't find areas that were specific to these really high cognitive functions, like the phrenologists believed. And so this kind of started this split in the field of, is aggregate view theory, which we'll talk about in a second, better than thinking about things in terms of localization? This, as we'll talk about in a second, is a really big thing that's still going on in neuroscience today. It's the idea of trying to understand whether the whole brain is necessary or whether very specific like regions are necessary. But what you need to kind of think about in terms of the debate at this point in time is that the localizationists believed that the higher cognitive functions were localizable, right? So as we get into kind of the cognitive neuroscience stuff that we will throughout the term, we will be talking a lot about localization. We'll be talking a lot about network properties, of how different brain regions cooperate and communicate with one another. But it's very different than it was at this point. So aggregate view theory, this kind of came from Florenz camp. He was like, no, these higher cognitive functions are way too complicated to just be in one spot. They require the whole brain, right? And you have, on the other side of this, the camp from the phrenology kind of way of looking at things was like, no, it's localizationable, it's localizable. And the aggregate view theory started to win out quite a bit, but then more evidence started to pour in about how there were very localizable things for very specific functions. So we have in 1836, this was, this physician was noticing that there were very similar speech deficits in these different people that had very similar lesions. And we'll talk about Broca and Wernicke that you guys probably know about in a minute. And John Hewlins Jackson, very prescient finding here. He noticed that when people were having epileptic seizures, that the seizure itself was progressing through different body points in different parts in time. And he proposed this idea that if the epileptic seizure is related to the activity that's going on in the brain, then there's probably a map of the body in the brain that seizure is working its way through, and that's why it's activating different regions at different times. This is something that is very true, that he was able to pick up on without the tools that we have today, which is really cool. And the other evidence that came in, like I mentioned, was Broca and Wernicke, right? These are findings that are talked about in every single intro psych class, right? And it was with Broca, he had a patient that he called Tan because that was all the person could say, but the person could understand everything you were saying to him. It was very specific to his ability to actually produce the speech, right? And Wernicke, on the other hand, noticed that a very different part of the brain was producing a different kind of deficit, right? So this one was kind of back in the parietal junction, back here, and he could talk freely, and the person actually felt like he was making sense, but nonsense came out of his mouth, just lots of words jumbled up together. And so around this time, there was kind of an impasse. Like, there wasn't really any tools yet to really dive into any of these ideas and to really like break apart who was right. And so we have, at the beginning of the 20th century, the technology is starting to allow us to start looking at smaller and smaller things. And we start to see the study of like the molecular things that are going on. And so Broadman is a name that you will actually hear a lot in cognitive neuroscience still today. He was one of the first people that tried to map out the brain and tried to look at different structures based on how the cells themselves were organized. So this guy, Franz Nissl, actually developed this staining technique where you could take a brain post-mortem and you could apply a stain to it, and the stain would allow you to see all of these different cell bodies. And so what Broadman did is he took sections from all of these different parts of the brain, and he looked at these Nissl stains, and he looked at how the organization of the neurons was different. You can see that they're all in these kind of layered forms here, but the layers themselves kind of differ from region to region. And he used this to create a map of 52 distinct regions based on what he called, what is considered cytoarchitecture. It's looking at the architecture of the cellular stuff that's going on. When you talk about how the brain is split up into different parts, the Broadman areas are brought up a lot. It's a map that a lot of neuroscientists still use to try to look like, if they're trying to understand attention is usually related to Broadman area 45. I just pulled that out of my head. That's not a real one, but that's kind of, they'll look in that specific region because that's where it's been found before. So Broadman will definitely come up again in the future. And then a new type of staining came out that just changed the game completely. And that was called the Golgi Method. It was a method that involved staining the neurons with silver chromate. And it was the first time ever that we could see individual cells, right? And up until this point, there was this belief that the brain and the nervous system was actually one big cell. There's this huge camp. This is right at the beginning of the 20th century. We still didn't know that the brain was composed of all of these individual billions of neurons. And everybody thought that it was just this glob of mixed up cytoplasm. But this technique allowed us to finally see these individual neurons. And this guy, Gall, which actually is the next slide, but this led to the neuron doctrine, which was the idea that the nervous system is made up of individual cells, right? And we knew before this, cell theory had already been developed. There were a lot of people that studied the different parts of the body and had noticed that like the integumentary system was built up of different skin cells, that the heart was made up of different cells, but it still prevailed late into the 1800s that the brain and the nervous system were an exception to that it wasn't different cells. Everything was all just kind of connected together. And that was because of this limitation of being able to actually get down and see at the really, really small molecular scale that there are gaps between these cells. And this guy, such a badass, Gall, used silver staining method, used the Golgi method, and just started drawing, started making all of these illustrations of all of these different neurons that he found. And he is really credited with being like the father of neuroscience, because he's what started to create this cascade of researchers that were like, okay, if the nervous system is individual cells, then we can study those individual components. We can look at those biological interactions. And his big contribution was that when he was able to get down small enough, he was able to see the axonal growth cone, which is the separation between the different cells. It's the first evidence that it wasn't just this big glob of stuff, and there's actually these individual things that were all cooperating, coordinating with one another. And got the Nobel Prize with Golgi, who had developed the same technique in the first place. This picture was one of his drawings, actually. So Charles Sherrington is one of the next guys that kind of added to the piece of the puzzle. He was a person that studied reflexes, right? And he noticed when he was timing these reflexes that if the nervous system was this continuous, uninterrupted thing, then the speed was wrong, right? It should be this continuous, uninterrupted thing, but that wasn't the case. And he's actually the one that coined the term synapse. He said, there's this separation point that's causing everything to slow down. But as you'll see in a minute, he was actually on the wrong side of the academic battle. Like, that's an honest description of it that would ensue in the 1930s. So we had kind of figured out that there's different cells, there's separation between those cells that is allowing for this communication, right? But what we didn't know is how the communication was happening across that gap, right? And so you have the soup versus spark debate. Super heated, 1930s. You have John Eccles, who was a student of Sherrington, who we just talked about, and he was in the camp of believing that all information in the brain was being transmitted electrically, right? He was in the spark camp. And you had this other group of researchers, Otto Lowy and Henry Dale, were starting to figure out that it's not that simple, that it's probably a chemical interaction that's happening after the electricity has happened. And this was huge. They were working with acetylcholine, which is a very prominent neurotransmitter in the peripheral nervous system. It's something they can pick up with big nerves that they were studying. And they actually went to blows at a conference, got into a fist fight over who was right about this. I mean, this was less than 100 years ago. People were punching each other in the face about whether it was electrical or chemical transmission at the synapse. Like, that's just like we take for granted how much we've figured out in the last 100 years. And the person that ended up winning, of course, was the soup part. But ironically, so John Eccles was the guy that was, this was the heated guy. When you really read about the interaction, John Eccles was just like, you're wrong, I'm right. And then John Eccles is the one that ends up finding out that he was wrong. And so he actually figures out, starts working with acetylcholine, like Otto Lowy and Henry Dale had been working with, and figures out that it is the acetylcholine that's causing the firing in the next cells. Right? And he developed Dale's law, which is based on the guy he punched in the face. And Dale's law was this idea that each individual neuron only produced one type of secretory chemical, one type of neurotransmitter that allowed it to communicate with the other cells. This isn't exactly true. It's mostly true. There are a lot of neurons that just produce one type of neurotransmitter, but there's a lot that produce multiple ones, as we'll kind of get into. We won't get a lot into it because our type of research is going to get into kind of the macro scale. But the discovery of glutamate, which is the most prevalent excitatory neurotransmitter in the body, and GABA, the most prominent inhibitory neurotransmitter, came really close, and serotonin was in there too. They started to kind of bathe these neurons in these different kinds of chemicals and started to see how the neurons then started to fire. And then what changed the game completely was the ability to genetically modify animals, which really you guys should not take for granted the fact that genetic modifications has changed the world we live in, right? There was this huge outflux of researchers. There had been this idea in academia that if you were a professor, you did not go to industry. That was no, that's against academic beliefs and our big ivory towers. But as soon as genetic modifications became possible, a ton of researchers started developing pharmaceuticals, started to be able to test these effects on these genetically modified rats. So the first people that did it were this group. It was Mintz and Janisch in 1974. They were able to successfully genetically modify these animals, but the modifications were not successful in being passed to the offspring. And so this other consortium of people, I'm not going to read all of their names. Please don't memorize a bunch of names, even the names that I've gone through. I'm not going to have you guys tested on these rich white guys that discovered stuff. But kind of this idea that I was just talking about, this really catapulted neuroscience in general, because now we were able to create behavioral deficits by specifically targeting and lesioning different areas of the brain genetically. It was huge advances. What we're not going to get into in this class is kind of what comes next in the neuroscience field. We're going to focus more on imaging tools and kind of the study of humans. But the stuff that molecular labs are able to do now is incredible. They can turn neurons on and off with light, with optogenetics. They can use dreads, which are targeted pharmaceuticals that only activate specific types of neurons. It's absolutely incredible what they're able to do. But like I mentioned earlier, a lot of neuroscience itself deals with animal models, right? So they're dealing with mice and rats and aplysia and zebrafish and all these kind of things. And they're making the assumption that based on what we talked about earlier, phylogenetics, right? We see this conservation of brain structures throughout evolution. So the idea with animal models is that if things are conserved as the organism gets more complex, then studying these simple animals can reveal things about how human cognition works, right? It's an assumption. It's something that is really hard to test. We can't do these kind of things on humans. So the other side of this, so kind of what we opened up with was the difference between the cognitive psychology side and the neuroscience side. So what we kind of just went through was the neuroscience side of things, right? The molecular side of things. And on the other side, you had this group that was studying psychology, and psychology was starting to become this big discipline, this rigorous scientific discipline. And these cognitive psychologists wanted to understand the brain as an information processing system, right? They knew that the brain itself was collecting information. They knew that there were probably complex processes that allowed for that information to be transformed and turned into these mental representations that we have. And so they kind of proposed this idea, kind of got ahead of myself, that cognition itself required these complex information building processes where you started with simple stuff that could be processed very quickly. And then as you got more complicated, the processing system is going to lag. It's going to take more time. And these mental representations, this is kind of just an example of what they were thinking at the time. So if I put this up on the board, and ask you guys to read it. Some of you can kind of probably work out some of the words, but it does not flow very well, right? Now, if I put this one up on the board, you can probably read through that really efficiently and really quickly, right? This is capitalizing on the fact that we have mental representations in our brain, that our brain is building a model of these individual words, right? Our brain knows what the length should be. Our brain knows what the first and last letters should be. And it uses those things to build up these representations. Now, the tool at the time that cognitive researchers had was reaction time, right? It was if a certain stimulus is given to the participant and we're requiring them to respond, right? So they view the stimulus, they have to process that stimulus. They have to make that mental representation, right? And that mental representation has to undergo transformations. And those transformations are then used to develop responses, to make decisions, right? And they believed that without any of the modern tools that we have to actually look at what the brain's doing, that they could understand something about these processes based on how long they were taking, right? So you have this guy, Franciscus Donders. This was like really, really early. You'll see that like the actual cognitive psychology was much later. It was more like the mid 1900s when this became a really used technique in the sciences. But it leads to this guy who is absolutely amazing. And if you guys have not met Mike Posner, he works here and he's one of the coolest people I've ever met. This is him getting a medal from Obama. He is a huge, huge pioneer in our field and was a huge pioneer in just the cognitive psychology side of things. And then ended up being one of the first people to ever do neuroimaging at all. And he's like right down the hall from me. It's really cool. So he developed, among others, this was one of the tasks that he set up where, oh, that's not good. Give me 1 second. Probably should have started with that. All right, cool. So he had this letter matching task, and he gave the participants very clear instructions that there would be 2 letters that would pop up on the screen, and the participant needed to respond in different conditions based on different things. Some of the conditions asked them if the 2 letters were identical. So they'd see a bunch of letters on the screen, tell me whether or not those letters are identical. They'd go through a bunch of trials, they would do that. And then in another condition, they would say, tell me whether or not those 2 letters are the same letter, right? They'd see uppercase letters, they'd see lowercase letters, identify if they're the same one. Another condition asked if they were both vowels. Another one if they were both consonants, and another one if they were different categories altogether. And what you can see in this graph was something that was amazing at the time for them to actually witness. This was mapping out the processing time that was required for these different types of categories. Just identifying whether or not these 2 letters were the same was a very easy process. Didn't require a lot of processing power. It could happen very quickly. If you were to identify that they were the same letter, okay, I need to bring up my representation of the letter A. It's got 2 different versions. Okay, cool, I can respond now. Took a little bit more time, right? Then as we get into these complex categories like vowels and consonants, well, we're bringing up very different types of representations. It's not just a representation of one letter. It's the representation of how the English alphabet works, right? What a vowel is, what a consonant is. That requires a lot more processing time, right? And so this was this window into the brain before we had actual windows into the brain. He created another one. This is still widely used even in neuroimaging tasks. It's the Posner cueing task. And it was used to try to see how attention was affecting the way that we process information, right? And so what he would do is he would put cues would come up on the screen. It would either be an arrow pointing in one direction, or it would be a box that's highlighting a certain area. And what that cue did was it told the participant where the object, in this case the star, was going to show up, probably, right? The cue on 80% of the trials pointed in the right direction. You're saying cue is going to show up here, the cue would pop up, and keep in mind that the subject is staring just at the fixation cross right here in the middle. And so he sees this, and he says, okay, it's probably going to show up there. It actually shows up, and then he hits the button, it was on the left, or it was on the right. But then he would throw in these trials where the cue would tell him that it was going to be in a certain direction, but then it wasn't. And what he found was that attention had these processing demands, right? If I get them to believe that it's going to be over here, I'm going to force their attention in that direction. I'm going to force the spotlight of their brain over there. And it's going to take them time, if that's the wrong spot, to reorient that attention and bring it somewhere else, right? This was way ahead of its time. And it seems like a very, very simple design, but it's getting at something really powerful, right? It's showing how attention is modulating these mental representations and these processes. So Saul Sternberg kind of followed this up in the memory domain, and he used reaction time again. This is one of the early cognitive tools, right? And he argued that 4 things need to happen when you're trying to remember something, right? You need to encode that information, so you need to store it somehow. You need to compare the representation to what was in the memory set. So basically they're shown these like 4 letters, and then they're shown 1 letter, and they say, was that letter in the set that you just saw, right? And so they have to encode all of this information. They're then showed this letter, and they have to bring this representation back up, right? And search that representation and say, okay, was the letter there or not? They have to make a decision, and then they have to respond. There are these different, very distinct processing steps that should each take a certain amount of time, right? And what you can see is that as the number of letters here got longer, the time to respond got extremely long too. And so Sternberg was arguing that these processes in the brain are serial. They need to happen one after another, right? And if you make one of those steps harder, it's going to slow the whole process down. And so this was kind of early in the cognitive camp, was this idea that is the brain processing things in this like hierarchical way? Is it processing the early stuff and then the more complicated stuff and then more complicated on top of that? Or is the brain able to process things in parallel? Is it able to hold multiple representations at the same time? And what this study with Riker showed was that there actually is this parallel processing going on. It was kind of a stint on the serial process that had come earlier. So he asked these participants if a certain letter was present in one of these words, right? And there were 3 different conditions. It was either an actual word, it was a nonsense word, or it was a bunch of X's with that letter in it. And what he found was that we're extremely accurate at determining if the letter was in the word, if it was an actual word. And so we're able to have this representation of the letter and this representation of the word being processed kind of at the same time. And it was the first evidence that there might be these multiple things going on at the same time. And this is really important when we start to get into the higher cognitive functions, because what you're going to see is that the brain is a massive parallel processor. There are certain regions of the brain that do process things serially. So these people were actually both right. There are very serial processes. The processing of sensory information goes from very simple sensory signals to build up perceptions of objects and things in our world. But there are also cases where one part of the brain might be focusing on the color of an object, while another part of the brain might be focusing on whether that object is moving or not, right? There's lots of things going on in parallel. Somehow all of that information is being combined to create this perceptual stream that we have. So that's just kind of reiterating that this is a parallel process. So this was kind of the last, or not the last, but this was one of the other big studies in cognitive psych was the Stroop task. You guys have probably all heard of the Stroop task. So you show the participants colored words, and the word itself is the name of a color, and the color either matches the word or it doesn't match the word, right? And what they found is that subjects are much worse at responding on these where there's a mismatch. And what it started to show, it was more evidence for this parallel processing idea, that we have a representation of color and we have a representation of the word. And those representations are actually competing with one another, and they're putting demands on our attention. And it was something that could be measured really early on, right? And so to kind of sum it up, we're going to get out a bit early today. This was kind of one of the simple lectures. Cognitive neuroscience is putting these two disciplines that we just talked about together, right? So you have cognitive psych, which is trying to understand the brain as a processing system, trying to understand these higher cognitive functions like attention and memory and competing representations and all these things. But it wasn't possible to look at the biological underpinnings until like the 90s. Now, EEG was there a lot earlier. There were a lot of people doing EEG at the beginning of the 1900s. But EEG didn't really start to become the dominant force that it was until a lot later. And a lot of that is because of computational demands. When we're talking about imaging, and what we'll kind of get into when we talk about methods, is that cognitive neuroscience is a huge data problem. We have tons and tons and tons of data that we have to analyze. Because we're looking at all of these different parts of the brain all at the same time, at multiple time points, in multiple people, right? And so before the 90s, before the 80s, we didn't have the computational power to be able to tackle problems like this, right? But we have now, that was kind of so MRI and PET. So PET came out in the 1980s and MRI was 1990s. And something that you'll see me kind of reiterate a lot throughout this class is that even though MRI was being done in the 90s, a lot of the early MRI studies are very limited. MRI is an extremely expensive thing to use. We just ran a study in my lab that collected, I think, 115 people, and we blew through $120,000 in less than a year, right? Very, very expensive tool to use. And because of that, when it first came out, most of these studies that are coming out only had like 15 people in them, right? You guys have all been through the stats sequence and all of that. How many people should you have in a study to have a good estimate of the average, right? You should have like 30 or more. That becomes even more complicated when you're doing MRI and when you're doing EEG, because the signals that we're picking up on are extremely messy. They're not clear. There's a lot of physiological stuff that's going on that's interfering with our ability to actually pick up on the signal that we're looking for. So when you're doing these studies that have not a lot of people, you really got to look at them with kind of an eye of skepticism, right? So this allowed cognition to be studied for the first time, kind of from a biological basis. And so this is going to set the stage for the rest of what we do now. And what I want to kind of get across to you guys, we're going to have a lecture next week on methods. Most of what happens in cognitive neuroscience is coming from MRI research. There was a study done in 2015 that showed that in 2014 there were 29,000 MRI publications and there were 5,000 EEG publications. And the other methods that we'll talk about didn't even hit the board, right? So this class is going to be very heavily focused, especially when we get to higher cognitive functions, on MRI and how we're using MRI. And when we get into the methods, I'm going to go a little bit deeper than the book does on understanding what MRI is telling us and how we're actually making sense of that data and what kind of things you can actually conclude when you read an MRI paper. There's a chapter in our book that says that we can read minds. That's complete bullshit. We'll get into why that's the case. But something I just want you guys to leave here with is an eye for skepticism, and just really, I want you guys to be able to look at papers and to say, okay, I trust this. They had good methods. They had a lot of people, that kind of things. Don't believe a lot of this popular science shit that I'll come across that says that we've figured out everything, because it's not the case. So that's it for today. I wish you guys go a little bit early. I'll see you on Tuesday for Tour of the Brain, or on Wednesday for Tour of the Brain.